This is a recording to show you how to do this activity for cell differentiation and gene expression. So first we're going to read through this reading here. In most human cells, the nucleus contains a full set of 23 pairs of chromosomes, which carry 20,000 to 25,000 genes. These genes are identical from cell to cell. So every cell has the same genes in it, and every cell has the same chromosomes in it. Through the process of protein synthesis with transcription and translation is how genes are transcribed to produce ribonucleic acid. The RNA is then translated to produce protein. If all cells in the same organism have the same genes, why don't they all make the same proteins? So this is our reminder that we're going to use this. There's going to be the same DNA in all cells, and that DNA is going to get transcribed to make RNA, and then the RNA is translated to make protein, and the protein does all the work of the cell. So some proteins are made by almost every cell because they're needed for basic cell function. Other proteins are only made by one type of cell or small groups of cells. Only white blood cells, for example, make antibodies, the proteins that help the body fight infection. Each of the more than 220 kinds of specialized cells in the human body make a characteristic group of proteins. Although the two human cells shown have the same genes in their nuclei, they are specialized to make different proteins. The lung cells are specialized for surfactant protein B, a protein found in the lung that's vital for reducing surface tension in the lungs and allowing the oxygen change in the alveoli to take place. The bone cell is a cell that's responsible for the breakdown of bone tissue and makes large amounts of the protein integrin. In each cell, only some of the genes are active or expressed. The activity of genes in a cell is called gene expression. In this activity, you will explore how some genes are turned on and off by molecules called transcription factors. These molecules control the transcription of DNA into RNA. So if you're looking at these pictures, this is one type of cell in your body. These are the lung cells, and this is another type of cell in your body. These are the bone cells. Both of these types of cells would have the same DNA in them. They have 46 chromosomes, so this 46 molecules of DNA. They have all the same genes, but they have very different functions because of the proteins that they make. So different areas of the DNA is red. So what we're going to try to find is how does the same set of genes direct the activity of 220 different types of human cells? So if you were in class, you would be working in a group of 4, and each person would be a different cell. That doesn't work as well as online, so we're going to have you just compare all 4 cells. It's really important that you notice the 4 types here. So one type of cell is the beta cell. The beta cell is in your pancreas, okay, which is right here in your midsection. The pancreas job is to produce the hormone insulin, and that regulates the cell's uptake and metabolism of sugar and fats. So the whole purpose of the pancreas in your body is making this insulin. The red blood cell is a different type of cell. Its job is to transport oxygen. So it makes hemoglobin, which is a protein that can carry oxygen to the other parts of the cell. So the red blood cell has to be able to fit through the arteries and capillaries and veins. It's got to be able to move through, and it's going to carry oxygen. The intestinal lining cells, so this is in your intestines, right, which is part of your digestive system. These are going to produce enzymes, which are proteins, that help to break down food. So they're going to help with the digestion of food. So that is their job. They're worried about digestion. And then the smooth muscle cells in your intestine. So again, in your intestine, you have muscle cells that are going to move the food along the digestive tract. Okay, so this is more about moving the food in the smooth muscle cells, where the intestinal lining cells is breaking down the food. So this picture here is called a karyotype. This is a picture of the 46 different chromosomes. So they come in sets of 2. So chromosome 1 has 2, chromosome 2 has 2, that are found in every single human cell. So this is with an XY, which is male, and an XX, which is female. So we're going to have you use the drawing tool, actually probably the shape tool, to draw a circle. And you are going to circle chromosome 2. I guess oval. It's not always the easiest. You might be able to move it afterwards. Let me see if I can't move the circle after I've drawn the circle. Maybe. Can I move it? I'm trying to circle chromosome 2. And I'm trying, there we go. And then I'm also trying to circle chromosome 11. I understand that this is a pain. Do your best. It's okay if it's not perfectly on there. So shape, circle, chromosome 11. You'll notice chromosome 11 is smaller than chromosome 2. They just put them in order from largest to smallest and then numbered them. Okay? So chromosome 1 is the biggest chromosome, 22 is the smallest, and then these are the sex chromosomes. Okay? So you'll notice that chromosome 2 in males and females is the same, chromosome 11 in males and females is the same. Now there might be different versions of the genes on there, but they're the same chromosomes. See if I can't delete that. I didn't mean to make that circle. All right, so you have circled chromosomes 2 and 11 in both. So now this part's really, really important. Anytime I'm talking about the genes, right here is the list of the genes in both chromosomes. And I want you to notice, do you notice right here it says chromosome 2? Here are the genes on chromosome 2. Okay, so ribosome protein S7, protein synthesis initiator, actin, cellular respiration enzyme, lactase, AGA enzyme. These are the proteins that are produced by the genes. So these are the genes. And then here's their functions. So later on in the conclusion, we're going to ask you about why certain cells turned on certain genes, and it depends on their function. Here's the names of the cells going down. So if you're looking at this column, here's what's turned on and turned off in the beta cell. This is what's turned on in chromosome 2. This is what's turned on in chromosome 11. Here's what's turned on in the red blood cell in chromosome 2 and in chromosome 11. Here's the intestinal lining cell, and here's the smooth muscle cell. This chart's super important. So what we're going to do is we're going to look at which genes are turned on in all of the cells. So we're going to look by gene. If it's turned on in all cells, what's turned off? So if it's got a minus sign, the DNA is still in the cell, but it's just not being read. Okay? And then if it's blue, it's specific to one type. So I am going to change the color. Let's see. So I want orange. And I'm going to draw an orange circle. Oh, there we go. On every one that has a plus. Oops, that was a terrible circle. Oh well, you get the idea. All the way across. It's helpful to start way over here. Drag your circle over. If there are 4 pluses going across the row, I'm doing an orange circle. Now look in this one. This one has minuses and then a plus, so I'm not going to do it in that one. I'm going to do it here. Anytime that there's 4 circles going across, what this is telling me is that this gene is found in it's turned on. First of all the genes are in all the cells, but this is turned on in every cell. I missed that plus, but you get the idea. This way. There we go. So if something's turned on in every cell, the function of it is probably a fairly generic or general function. Whereas if it's turned on in only one cell, it's probably a very specific function. So if you look at the one I'm doing right now, DNA repair protein, that's found in every cell. Why would it be found in every cell? Because every cell needs to repair the damage to their DNA. Cellular respiration enzyme, that's found in every cell. Why is it in every cell? Because every cell needs to do cellular respiration. So now I have put the orange circles in every single one that is on in all the cells. Now I'm going to look for the genes that are repressed or turned off in certain cells. So I'm going to switch this to black. And I'm going to do every single minus. Okay. Oops, I'm kind of missing. That was a fail. Maybe start over here. There you go. So all of the minuses, whether there's 3 of them or 4 of them across, you're circling in black. And then here we go. So I'm doing both chromosome 2 and chromosome 11. So this is showing me which ones are always turned off, like they are never on. So what's happening here is that the beta cell is never ever going to turn on the actin smooth muscle gene. It contains that gene, but it doesn't need to use it. And if you look at it, that gene is about cell movement. The beta cell is about making insulin. It doesn't need to move. Okay? And it's not about like the red blood cell moving through the, it's about like active movement, like a muscle. The AGA enzyme is for like your liver breaks down toxic substances. None of these are liver cells, so it's often all of these. This last circle I'm going to do is the blue circle. So I'm going to go here. And this blue one, this is a gene that's only on in specific types of cells. So its function is probably going to be specific to the type of cell that it's on in. I feel like I've missed one. Oh, here it is. Okay. So if you look at this, actin smooth muscle type, this is a gene that is found in every single cell, but it's only on in the muscle cells. And if you think about the function of a muscle is for movement. Lactase is an enzyme that breaks down food. Look at that. It's on in the intestinal lining cells, and the intestine's job is to break down food. So this enzyme is turned on there. The gene is in all of the cells, but it's only on here. Hemoglobin B is on in the red blood cells. Its job is to carry oxygen. The red blood cell's job is to carry oxygen. It's on in that cell. Insulin is on in the beta cell. It is a hormone that metabolizes sugar. That's why it's on in the pancreas cells that make insulin. All right, so now we have this map, this chart is the best. This shows you very easily what the genes are and what's on and what. But now we're going to go to like a physical map of a chromosome. This might be a little tricky. We're going to have to go up and down. What I need you to notice right here is that the cell types are across the top. That could be easy to miss. So this is a picture of chromosome 2 for the beta cell and chromosome 2 for the red blood cell, chromosome 2 for the intestinal lining cell, and for the smooth muscle cell. This is chromosome 11 for those cells. You'll notice that I have the genes in the different positions on those chromosomes. So a single chromosome can have multiple genes on it. Now, what I would like for you to do is I want you to draw a black line across the genes that are never expressed. So what I'm going to do is I'm going to look on here and I'm going to notice that actin, lactase, and AGA are off in the first one. So I'm going to go to my shape tool and I want to do a line and I want to do it in black. And I'm going to go to actin. Can I make this thicker? Oh, up here. I'm going to change this thickness to maybe 6. So to actin, I'm going to put a black line right there because that's off in this one. And then I'm going to go to lactase and I'm going to go to AGA. Okay? So that is off because they're off here. Then I'm going to see that hemoglobin is off in this one too. So I'm going to go to hemoglobin, which is way up here. Do you see that it says hemoglobin right there? That's how I figured that out. Then for the next one, I am going to go, so you're basically just coloring your map as it looks up here. So actin, lactase, and AGA are off in this one. I'm just going to do all the blacks first. So actin, lactase, AGA. I missed this line. There you go. And then this is a red blood cell, so it's definitely not hemoglobin. What does this one have off? It has insulin. Insulin is off here. Oh, insulin's the lower line. Okay. Then I'm going to come up here, and actin and AGA are off, and then hemoglobin and insulin. So actin, AGA, hemoglobin, and insulin. They're next to each other. And then my last one, I'm looking for these lines, lactase and AGA, and hemoglobin and insulin. Lactase and AGA, and hemoglobin and insulin. So if you look at these, so this is the chromosome map of the pancreas, this is the red blood cell, this is the intestinal lining cell, this is a smooth muscle cell. I'll notice as I look across, one of these is not like the other. So one of these has this one on, like this is going to be on in this one, but not in the others. So actin is like the special one. So I'm going to go to blue. I'm going to stay on line, but go to blue. And the one that's like the special one, so there you go. So this one here, so these three are off, so this is going to be on here. Like I could look back at the chart, but this is how this works. So this tells me that lactase is the special enzyme for intestinal lining cell. Actin is the special enzyme for smooth muscle cell. Okay, this tells me hemoglobin is the special enzyme here for the red blood cell. And insulin is the special enzyme here for the beta cell. Okay, so that's how we're doing this here. Then all the other ones are going to be on all the way across. That tells me that these are very general functions that are needed. Okay, so I'm going to change this color to orange, right? Because orange is what we're doing. And so I'm just going to draw these lines across. So everything that's all the way across. So ribosome protein S7. That is a protein that's on in all of these because it makes ribosomes, and all cells need ribosomes. Ribosomes make protein. Protein synthesis initiator. That's on in all cells because all cells need to initiate protein synthesis, making proteins. Okay? Cell growth controller. That's going to be on. Oops, I did a terrible job with that line. It's going to be on in all the cells because all cells have to control their cell growth. Fat and protein breakdown enzyme. That's going to be on in all cells because all cells will need to break down fats and proteins. And DNA repair will be on in all cells because all cells will need to repair their DNA. So looking at these chromosome maps, I can see things that are the same and things that are different. But I notice that the actual chromosomes are the same. The DNA molecule is the same in every cell. The genes are all present. If there's a black line, it doesn't mean it's missing. It just means it's turned off. So the DNA is actually really tightly coiled there. So it's off. Oops, I missed one. I missed cellular respiration enzyme. It's right here. Okay, so yours should look like this. So now you can compare the pancreas to the red blood cell, to the intestinal lining cell, to the smooth muscle cell. So now we're down to cellular events. So there's two main concepts you need to get out of this lab. One concept is that every cell has the same DNA and the same genes, but they're not all turned on the same. So you can look at this picture and see that certain cells have certain genes turned off. The other concept you need to understand is that even if a cell has the ability to turn on or off a gene, or like these ones in orange or blue, these are ones that they typically would potentially turn on and off. They don't always turn it on and off. Okay? So if you look at fat and protein breakdown enzyme, you can turn that on, but you'd be useful to only turn it on when there's actually fat and protein present. If it's not there, then you wouldn't have it. Okay? So ribosome proteins, you would turn that on when you need more ribosomes. If you have enough ribosomes, you could turn it off. So we're going to look at events. Events are things that are going to happen to the cell that are going to cause the genes to want to turn on and off. So the black lines are already present here. So the black lines are things that you will never change. What you're going to change is you are going to put now a green line if the gene is being turned on because of the event, and a red line if it's being turned off. Okay, I will do the first two, but then you'll have to do the last three on your own, just so that you get the idea. So the event after a full meal of protein and fat. So the body is eating protein and fat. Now the different cells are going to be affected by that. So a person's body is going to need more enzymes to break down fat and proteins. Add a green line to the gene for fat and protein breakdown enzymes in all cells. So this is going to affect all cells. So first thing you have to do is find fat and protein breakdown enzyme and believe that that exists. I'm sorry that this is really small. Here is fat and protein breakdown enzyme. We are going to activate that in all cells. So by putting a green line, we're saying we're turning it on. So green now means on. So what happens is that if the cell does not have an activator, it might make that protein just really, really slowly. But if you put this green activator on, you're saying, hey, it's time to speed this process up, and we're going to make a lot more of this protein. So remember that I'm activating the DNA, because chromosome's the DNA. That means it's going to get transcribed to RNA and then translated to protein. So we're now opening up this section of the DNA to be read in all of these cells, and all of these cells are going to make this protein. So now that was our first event. We ate fat and protein. Second event, our developing red blood cells must make a large amount of hemoglobin. Draw 2 green lines on the gene for hemoglobin in the red blood cell only. So now I need to read up here on these gray things, which one's my red blood cell? It's just the middle one. So draw 2 green lines on hemoglobin. So hemoglobin is down here. That's going to be hard to draw 2 green lines, but I made them really thick. There we go. So I'm drawing 2 green lines on hemoglobin, and what I want you to notice is that this event only affected one type of specialized cell. So you need to do the next 3 on your own. I'm not going to do all of them for you. Okay? So the intestinal lining cell and maturing red blood cell do not need to divide anymore. Add a green line to the cell cycle control gene, which will produce a protein that prevents them from dividing. So realize I'm only talking about 2 types of cells, intestinal lining cell and red blood cell. So you're only going to work on those 2 cells, okay? And follow these directions for those 2 cells, not for all of the cells. For number 4, there's milk, which contains lactose in the small intestine. So again, intestinal lining cells. The lactase gene is expressed in the intestinal lining cells. Add a green activator, that means to put a green line. This will increase production of the lactase enzyme. So draw a green line on the lactase gene. And then cell event 5, the pancreatic beta cell has enough insulin at a red repressor. That means draw a red line. Draw a red line to the insulin gene for the beta cell. So again, just looking at the beta cell, draw the red repressor. So now for our analysis. Compare the chromosomes in the 4 types of cells. Are chromosomes 2 and 11 in the muscle cells the same as chromosomes 2 and 11 in the other cells? So when you look at, you can just even look at this picture, when you look at the chromosomes, do all of the cells have the same chromosomes? Do they all have chromosomes 2 and 11? And the next question is going to be, do they all have the same genes? We're not asking whether they're turned on or not, off or not. Do they all have chromosome 2? Do they all have chromosome 11? Do they all have the same genes? So question 1 asks you, do they all have the same chromosomes? Question 2 asks you, do they all have the same genes? Question 3 says, how was the expression of the genes different in the different cells? Which genes were on and which genes were off? So go back to either this picture here, this picture helps you to see, or go to the chart. Okay, so again, the orange means that it's on in like all of the cells. blue is also on, and black means off. So compare, so in here you're comparing down a column. Compare the different types of cells, comparing, and here's your genes, which ones are on and off. Okay? So the question is, was there different expression of genes? Did some have some on and some have different ones off? What kind of genes were permanently inactivated in red blood cells, and why were they inactivated? So let's go back to that chart. Sorry, lots of scrolling. So when you go to red blood cells, that's the second column. That's what this one here. So what's permanently off? So I'm looking for the black ones here, and I'm going to look at the functions of the black genes. So actin, lactase, AGA, insulin. And then I'm going to go back up here and look at the function of the red blood cell. And that will help me to know why they might be off in that cell. Explain why some proteins are made by nearly all cells. Give 2 examples of genes turned on all cells. So go back to that chart, look at the ones that are orange all the way across. Give me the names of 2 of those genes, and tell me why. Look at their functions. What types of events caused short-term changes? So come up here to the cells events and tell me about some of the things that happened in the body. You don't need to do all 5, maybe give me 2. And then lastly, pick one type of cell and explain how gene expression is related to the cell's ability to perform its function in the body. So again, think up here, what's the function of the 4 types of cells? And then think on here, specifically looking at the specific genes that are turned on just one, how would the expression of that type of gene give it a special function? Please explain that.